Good afternoon, Kendall. How are you? Good. Good. I was just checking my temperatures. It's trying to warm up out there. It's supposed to get up to 70 degrees, but it's still cold to me. Not there yet. Good afternoon, everybody. Good afternoon. Good afternoon. Hope everybody had a good weekend. While we're waiting for folks to come in, I let me look and see how many people. A few of you have turned in the worksheet. The pedigree video wrap up. But I asked you not to finish it. So what we're going to do is finish it today. First, show you a picture of what we have. I think only a couple of you have done it. Okay. And what I did, instead of creating a new folder for the second page, is I have extended the due date on this folder so that you can take this worksheet back and then finish it. And then I've changed the total to incorporate the second page. And I did that the other day because I didn't think you were going to finish the whole page. See one. All right, Jacob, you've done it. Kendall, you've done it. Who else is here? Michaela, you haven't, so you hang on to yours. Sorry, Malia, not Michaela, Malia. All right, so if you've already submitted the worksheet, which Kendall and Jacob have done, find it again. You're going to resubmit it. I think I might have to click on resubmit for you folks so that you can do that again. But we're going to finish it. So I'm going to wait for everybody to come in. Okay, Andrea, she submitted hers. Good afternoon, Andrea. I was just explaining to them that rather than submit the worksheet again twice, I'm just going to let you all take the first one back and then resubmit the completed worksheet in the same folder. Before I have to click on anything, just see if you can do that. If not, I'll change it later. But I have changed the due date to this week so that we can finish that worksheet. So let me pull up my share for a minute. Wow, everybody's, because we're still missing half the class. All right, and I do want to keep moving forward because we do have a lot to cover since you all have an end of course test to take. Wednesdays, we're supposed to do those practice questions. So we really only have the 2 days a week to work on new material. So we are going to try to keep pushing forward um, a little bit. All right. Good afternoon, Nikila. All right. So again, the worksheet that you all had on the pedigree recap, and I pulled this video. I'm not showing the whole video. I'm just showing you these pictures. The question in the box for number 16 that you had to answer. I just wanted to make sure, since everybody's taking their paper back, just make sure that you've done it correctly. So in that box 16, you should have 3 generations, the way they have it in this diagram, except you are using the letter T. So you're using a capital T for someone who is able to taste the chemical T PTC, and a lowercase t for someone who has the inability to taste it. So in that box for 16, you were diagramming a father and mother, and they have 3 children. It says that the father and mother have the ability to taste. But based on your chart above, there's 2 things they could be. They could be heterozygous or homozygous for that trait. So you have to decide, based on the fact that they have children who both have the ability to taste and the inability to taste, you have to decide what the parents should be. And considering that the only way, hold on, let somebody else in. Considering that the only way that you can have a daughter who is a non-taster is if she gets 1 gene from the mom and 1 gene from the dad. So that means that both parents who are tasters have to be heterozygous. They have to be big T, little t, both parents. Okay. The way you're supposed to draw this, you're supposed to put shading for individuals who have the trait. So that means the daughter should be shaded. You need to put the initials outside the box if you're shading, because you need to show the initials. So you need to make sure that the shading doesn't cover up your initials, or you can, some people are just shading around the initials, but the box or the circle shouldn't be totally shaded in so that I can't see the letters. The 2 sons have a choice because they could have gotten both capital T's from mom and dad, or they could get one of each from their parents. So that you need to indicate that you're not quite sure what the sons are going to be, because they could be either or, but for sure they're going to have a capital T because they are tasters. All right, then you have to show the same way. So in this case, um, like the one up here, number 8 would have been the circle with the daughter. So they'd switch places because they're saying the daughter got married and she married someone who's also a non-taster. So you have to uh, connect the line from circle to square for representing the daughter and her husband, and then a line going down, and they had 2 sons. So you're going to draw 2 squares, and if the being as they're both tasters and they only have 1 choice to give, so you have to indicate that, and there should be 2 squares for her, for them. All right, now the next thing I want to talk about is, so some of you have already done it, so I'm just reviewing that, so since you're taking your papers back, that you make sure that you did it correctly. All right, the second part of the question of the paper, number 20, gives you a sex-linked activity, and you have to figure out what it is. Okay, so let me move this along just a little bit. Okay, well, it's more like the first because it's simple. But in this case, you have to, you're attaching the subscript for, they're using a lowercase b for color blindness. Okay. All right. So the second part, so 18, 19, 20, and so on are for color blindness. So the B, capital B means that they can see colors. Lowercase b says they do not. Okay. Again, remember if it's shaded, they have the characteristic. If it's not shaded, they don't. So that based on that, you have to figure out what 20 and 21 are talking about, and 22. Then at the bottom, you have a different pedigree to look at. You're back to looking at autosomal. And remember, autosomal traits are the ones dealing with the body. They are not sex linked. So then you have another pedigree to fill out there. So what I want you to do, and I do want everybody to, because most of you have finished with the first page, so this shouldn't take you as long. So I want to give 30 minutes for you to complete this page. Let me know if I need to resubmit, resubmit the assign or reassign the paper. For those of you who already submitted, let me know if I have to reassign it to you in order for you to use the same folder to resubmit the completed page. Okay, so again, most of you already did 1 through 19, which is what I asked you to do. And now I want you to finish the paper, but I want you to use the same folder. Okay, so again, I want you to use the next 30 minutes to work on that. Okay, trying to get sure. All right, so what I'm talking about for you, okay, so everybody should have done that. So you're working on, everybody should be working on this page here. Okay, so again, that's just a couple of questions. You should have finished this part already. If you haven't, it's due. But work on that and just work on these last few questions and submit. So 30 minutes, 30 minutes. It's about 104. And then we're going to move on. Oops. Can you please stop jumping? All right, so we have one more lesson in this module to do, and then we have the review. So I want you to go ahead and if you finish this one early, I want you to look at karyotypes. That's the next activity that we have to do. We have to be able to read a karyotype map and be able to map chromosomes. So if you finish this and submit it today, which you need to do in the next 30 minutes, go to this page in Canvas. All right, I'm going to come out so I can take attendance. All right, so again, if you are logged in, that's what you need to be doing. When you logged into class, please do the work that for this class so that we can stay current and on top of things. As right now, some of you are behind. We are coming up on progress reports, so you need to make sure that any outstanding assignments that you might have are caught up by the end of this week. Okay, Andrea, I'm just seeing your question. Hopefully I had answered it earlier. All right, again, please let me know if you are not able to resubmit that document in the same folder without me reassigning it. Those of you who had already submitted, those of you who haven't, you don't have that problem. All right, and again, if you finish that, um, go on to the karyotype. I'm about to pull up the karyotype activity for you to look at. So please read the page on karyotypes, the next page. That is our last unit in Canvas before we test this unit. So again, a good thing to do would be to, if you have time, is to go back and look at the vocabulary. Lots of vocabulary with this unit. Mm. Yes, please. Yes, so when you're done with the worksheet, um, submit it as a reattempt, and it should let you, but if it doesn't, let me know and I'll go in and reassign. I haven't reassigned them yet, but if I need to reassign it, I will. So let me know if you can do that without me. You should have multiple attempts. So do it as a reattempt, and it should cancel out the first submission. All right, folks, we got another 10 minutes. Again, I'm about to show a short video on karyotyping. So if you are finished and you've uploaded your worksheet, the completed worksheet, please read the next page on karyotypes. Ooh. All right, Kendall, did it let you? Because right now I'm looking at the same original page. I'm just going to go ahead. I'm just going to go ahead and reassign everybody. All right, and folks, you got to remember that you are only using one letter, so we don't turn around and use a second letter. Like Kayla, I'm looking at yours. All of a sudden, you went from using T's to using H's. I don't know where the H came from, but there should be no H's. I do not, Kendall, but I will ask at the meeting tomorrow. What grade are you in? Okay, I will check. My list of questions. Okay, I'm gonna ask y'all to stop for a few minutes and watch this short video on karyotypes.
I've seen too many pictures of chromosomes. Well, diagrams, really. Diagrams are awesome, but I've learned I need to be careful about misconceptions that can come with them. See, in diagrams, I've seen chromosomes typically drawn in an X shape. So when I was younger, I thought, okay, all chromosomes are an X shape, which is really confusing when I heard people could have two X chromosomes or an X and a Y chromosome, because then I started to wonder, why are these chromosomes resembling the letters of the alphabet? And then sometimes they look like single stick pieces and not X shapes, and I wondered what is going on with these things. I also thought that if humans have 46 chromosomes, then they're in there somewhere deep inside the body, that if you need to analyze them, you'd have to find them and make sure not to do anything to them because you only have 46 of them. Well, these are some misconceptions I had. We're hoping to clear up a little bit about chromosomes. Now, our illustrations are not scientific, obviously, but they can help with our explanations, which are. While you can find chromosomes in bacteria where the chromosomes tend to have circular shapes, we are going to focus on eukaryote chromosomes. Specifically, we're going to focus on human chromosomes. Chromosomes are made of chromatin, and chromatin consists of DNA and protein. A chromosome itself is really intense packaging. It starts with DNA being wound around proteins called histones. This forms nucleosomes, which are often considered to look like beads. More and more condensing packaging levels occur. We highly encourage you to explore this until it results into the chromosome level. But why all this packing? Well, in your cells, you have a lot of DNA and it has to fit inside a nucleus. Another reason it's also really important for when you make more cells, like in the process of mitosis. But in mitosis, you have to be able to move DNA into new daughter cells. You have so much DNA that these highly packed chromosome units make it easier to do so. By the way, it's not always packed up so tightly. For example, in DNA replication, DNA typically needs to be unwound. There are a variety of factors that also influence how tightly packed chromatin is. Let's get kind of a general idea about how human chromosomes may be represented. Human chromosomes can be like this as a single component, or they can be replicated, like this. In our chromosome counting video, we mentioned when chromosomes are duplicated during interphase, the newly made chromosome is still bound here in this region called the centromere. By the way, this region does not technically have to be in the center of the chromosome. Because of it being joined here, we still count this as one chromosome unit. But that can be kind of confusing. We say that there are two sister chromatids. So one chromosome here, it duplicates, and now, while this is still attached, it's still one chromosome. So in humans, you have 46 chromosomes. In interphase, they are duplicated. There are still 46 chromosomes here, but that's 92 chromatids in the picture. During a stage of mitosis, those 92 chromatids get pulled apart, so each cell will have 46 chromosomes. Now, just a side note, while most of your body cells have 46 chromosomes, it's important to remember that certain genes on the chromosome may be turned on or off by a variety of factors. For example, you wouldn't want your eye cells actively using a gene to produce stomach enzymes. That'd be a problem. So, would you ever get a chance to see all 46 of your chromosomes clearly? Well, you could in something called a karyotype, an image of all your chromosomes. It's possible to have a karyotype made as part of some types of medical screenings. In a karyotype, the chromosomes are stained and visible. The chromosomes from a karyotype are typically from a cell that is in a mitosis stage, typically metaphase. As chromosomes are condensed and thick, a karyotype during interphase would be a challenge because you don't have that condensed DNA packing. Now, something confusing to clarify. When searching for karyotypes, sometimes it'll be represented kind of like this diagram, and sometimes you see something like this. And while both show 46 chromosomes, the number of chromatids in the two images here seem different, so what's going on? Now, while the mitosis stage is anaphase or telophase would be a time when each chromosome would have a single chromatid, those phases are generally not when you do a typical karyotype. For optimal viewing, a typical karyotype is taken at or right before metaphase. Recall from mitosis that in metaphase, the chromosome would have two joined sister chromatids. It's just that many times the joined sister chromatids of the chromosome can be so close together that to an untrained eye, it might be difficult to see that each chromosome in the karyotype technically has two sister chromatids. When arranging the karyotype, the chromosomes are arranged in homologous pairs. Homologous chromosomes are about the same size and contain the same types of genes, and in each homologous pair, you receive one chromosome from one parent and one chromosome from the other parent. 23 pairs. 22 of the pairs, 44 of the chromosomes, are called autosomes. That means they are not related to your biological sex. They may have genes related to eye color or height, or hair texture. The last two chromosomes are called sex chromosomes because they determine biological sex. Females have two X chromosomes, males have one X and one Y chromosome. Oh, and just a side note, the name X and the name Y have nothing to do with the shape of them. The reason is an interesting topic to look up, though. So 23 of these chromosomes are from the father, from a sperm cell, which is a gamete. Unlike body cells, gametes have the number of chromosomes as body cells. Sperm cells are considered haploid because they only have one set of chromosomes. The other 23 of the chromosomes came from the mother, contained in an egg, which is also a gamete. An egg cell is haploid because, like the sperm cell, it contains only one set of chromosomes. When a sperm and egg cell combine, they form a fertilized egg, known as a zygote. The resulting cell is diploid, as it contains two sets of chromosomes, and it will divide to continue to form more diploid cells. You are a diploid organism. Now, if you look at the sex chromosomes, you know that mothers can only give an X chromosome, because that's the only type of sex chromosome they have. But fathers could give an X, the baby would be XX, female, or a Y, the baby is XY, male. So fathers determine the biological sex. So, why do we care about karyotypes or learning about chromosomes? Well, one reason is it can really help when we are trying to understand genetic disorders. A karyotype can reveal missing chromosomes or extra chromosomes at specific locations. Do you have an interest in a career that involves this area of study? The field of genetics continues to expand, and the career of a genetic counselor may be something to read up on. Well, that's it for the Amoeba Sisters, and we remind you to stay curious.
All right. In general, in addition to showing you what a karyotype looks like, that was a great review video. Okay, so make sure you've done that karyotype. Make sure you uploaded your pedigree. And I've put in a new worksheet, which we'll look at on Wednesday a little bit, because Wednesday we're supposed to do those progress learning activities as well, but we won't spend the whole time doing that. So, karyotypes. Why did it jump? Karyotypes. All right. So, what we're going to look at the rest of the week are these mutations that occur from having, so they showed you that you're supposed to have 2 pairs of everything. Occasionally, they have maybe just 1 chromosome, so that creates one type of disease. You have maybe 3 chromosomes where you should have 2, and that creates a different type of disease. So we're going to look at the different diseases that occur when you have what they call trisomy. This is trisomy. This is what a normal one looks like. So if you haven't read this page, go through and read that page. And again, I've put the assignment in. So let me pull that up. Oh, that's not what I want. I want this. All right, so it'll say karyotyping. And I don't expect you to start it today, but it's in the folder for you to look at. But I'm showing it to you today. All right, so on the worksheet for karyotype, again, the video showed you that what they do is they map all of your chromosomes. So a karyotype is just a map of all the chromosomes. And they, instead of them being scattered all over the place, they put them together in matching pairs. Okay, so these are all matching, different types of matching pairs. So up here at the top, the question wants you to circle autosomal. So again, you need to know that the vocabulary word autosomal means all the body ones. And then put a box around the sex chromosome. So basically, you're going to circle everything except these last 2 over here in the corner. You're going to put a box around those because those are the X and Y. It tells you down, I know it's very small, but it says X and Y. So it says 20, they number everything 1 to 22, and then it says X and Y. So those are the sex chromosomes. Everything else is an autosomal. And because this one has an X and a Y, they're asking you, is it male or female? And you're supposed to say it's male because males are X and Y. If they had said 2 X chromosomes, it would have been female. All right. And again, normal, it says how many chromosomes. It doesn't say how many pairs. It says how many chromosomes does a normal person have? They have 46. So this picture shows 23 pairs, but you count them one at a time, there are 46 chromosomes. It says put a star on a homologous pair. So you can take the first one and put a star on it because those are homologous, meaning they code for the same genes. That's what homologous, homo means same. So that means that both of those genes, one from mom, one from dad, have the same characteristics on it. That's what we mean by a homologous pair of chromosomes. Okay. Then it says, look at the karyotype to the right. Do you see any chromosomal abnormalities? So they mean this second set down here to the bottom. All right, and I'm going to tell you, if you look very carefully, the last pair, instead of it being X and Y, you've got 3 X's. You're only supposed to have 2. So there is an abnormality. So you want to say yes. And then later on down here, they explain to you what all of the different abnormalities are. All right, so I'm going to stop there and we'll continue this discussion after I know that you have read the page, because we do need to read that page. So, nope, I don't want that. I want this. Okay, so I do want to make sure that you have read this. We'll play this video and we will go through answering these questions here. And again, this is the last module unit in the module, so we'll be looking to wrap things up all together before by next week. This week is gonna go has a lot to do. Okay, so just a heads up. All right, if you have no questions for me, make sure that you get those other worksheets submitted and we're good to go.